Hi, my name is Krista and I've just finished my second year studying biology at the University of Bristol, and for the last 7 weeks I've been working here at the University of Oxford with Dr. Mariana Schuster on her project, the proteolysis of plant immune receptors. Now, I decided to apply for this project as at the start of my second year I noticed that my interest in plant sciences was really starting to blossom, as I was really interested in plant cellular mechanisms and plant pathogen interactions, and then how this links to food security on the whole. So I applied to this project after one of my lecturers sent round the link to the BSPP vacation schemes, and then I was accepted and let onto the project with Mariana Schuster. So that's what I've been doing for the last 7 weeks, and I'll tell you a little bit more about the project now.
The primary aim of our study was to validate in planta the proteolysis of plant membrane immune receptor proteins. Plant membrane immune receptor proteins are essential for the detection and affecting intracellular responses to pathogens. Sometimes, with or without the presence of a pathogen, our receptor proteins can undergo proteolysis. This proteolysis can lead to the repression of a protein or the formation of proteoforms with potential new functions.
So, what were we actually doing in the lab? During our study, we hoped to observe this proteolysis in our model plants. This meant we needed to validate our candidates before expressing them via transient expression.
So here I am in the greenhouse collecting some plants for us to use later. But before we did any of this, we had to create our plasmids. So we created our plasmids via Gibson assembly and then transformed them into E. coli for cloning. Following mini preps and sequencing, we transformed our plasmids into A. tumeficiens so then we could perform agroinfiltration into our N-bentharmiana plants. Now, here I am with the leaves that have undergone agroinfiltration. It's also worth mentioning that our plasmids were marked with a GFP tag, which meant we were able to visualize any protein expression using the Typhoon scanner. Here is one of our scans. You can see that in plasmids 20 and 22, the GFP is very visible and expressed in planta. So here I am actually using the Typhoon scanner, which made the image I've just shown you. The image itself did have to be edited using ImageJ software, as the Typhoon scanner only produces the images in black and white. But following these successful transient transformations, we then extracted the leaf tissue and froze it in liquid nitrogen, which is what I'm doing here. This can be used in the future in Western blot, SDS page, Kumasi staining, which means we can then visualize our proteoforms and determine whether shedding has occurred.
So far, we have found evidence that shedding events are occurring in membrane receptor proteins. Unfortunately, due to time constraints, I've had to leave the project while it's still in its infancy and won't be able to work further in this very interesting field of research, at least for the time being. I will say, however, that I've really engaged with the work here and I think the experience was really valuable. I would recommend to anyone studying currently to consider the BSPP vacation bursaries and apply, because it's definitely something worth doing.